Hello students, today we are taking our next topic from the chapter Biological Classification. What is that? Reproduction in fungi. You can call it as fungi, you can call it as fungi, all the pronunciation are correct. But simply you should know we are talking about one of the kingdoms, right? So we have to discuss the reproduction, the means of reproduction in this kingdom, right? So our main focus will be there. Now let's take the topic where we will discuss the 5 kingdom classification. Earlier, even in one of my videos, I discussed about the 5 kingdom classification given by Whitaker, where first kingdom is the Monera, after that is the Protista, after that is fungi, which is our today's topic for our discussion, then Plantae, then Animalia, right? So, which is our point of discussion today? That will be the kingdom fungi, right? That is our point of discussion. Okay, now let's talk about first of all the journal characters for the kingdom fungi so that we can enter into our today's topic, that is regarding the reproduction. Let's talk a little bit about its introduction. Can I say this is a type of the unique type of the kingdom? Why it is so much unique? Because it is made up of that heterotrophic organism, right? Being it is heterotrophic, all organisms here are heterotrophic, right? Which can be saprophytic, which can be parasitic, which can be even showing symbiotic association. They will show you the diversity. This is very important question in its introduction. It will show diversity in its habitat and even its morphology. If I go with its habitat, the word given in your book is that they are cosmopolitan. Cosmopolitan means it is present everywhere, right? So everywhere means there is no country who do not know about what is fungus. So on that ground, this is a cosmopolitan, which can be present in the air, which can be present in water, and even it can present in soil, and even on the body of animals and plants, like a pathogens, right? And morphologically, why it is diverse? Because you will find they are showing the unicellular creature here, which is no doubt is very less rarely. For example, yeast. But this is very common. Yeast is common, right? But being unicellular is not common in this kingdom. They are showing the filamentous form structures, which are cylindrical elongated ferns. But that filamentous structures, you call them as technically as hyphae. Hyphae is a plural, hypha is a singular of that. Now here what is that? These hyphae can be septate or it can be aseptate, right? Septate means these cylindrical structures, or I must say elongated structures, which are, they are having the septa in between. Means the cross walls are there. If cross walls are present, can I say it become the multicellular structures? Aseptate means they are simply elongated cylinder. The word of cylinder is SCLENDR, both for here, even it is cylinder, means elongated. Like this marker is there, it is elongated and rounded. This you call it as a cylinder form. Now septate and aseptate, but both are cylinder and elongated filamentous form. If cross walls are there, you call it as septate. If not there, many nuclei will be present. Such type of hyphae, even you call it as cynocytic hyphae, right? I should write here cynocytic, right? So cynocytic hyphae you will find where the septa is not present, and you will find multinucleated structure. Whereas where septa is present, it is going to be multicellular, with every cell is having one nucleus, and even they are having uniqueness in their cell wall composition. Its cell wall is made up of chitin. Chitin you will learn in the chapter biomolecules. This is the homopolysaccharide, right, which is made up of the complex sugars. The monomers are the complex sugar. Do remember the word chitin, right? So it is having a cell wall composition, unique as well, chitin here. You will learn the chitin is even present in exoskeleton of arthropods in biomolecules. But right now, even in fungus, their cell wall is made up of chitin, right? So this is somewhat about it, journal information. And even one more point let me to take, being heterotrophic, the reserve food material is in a form of the glycogen. Reserve food material is in a form of glycogen, right? Being heterotrophic, even oil droplets can be there, right? Okay, now coming to the back to the main topic, reproduction. Now in the reproduction, they are showing in the means of reproduction, major three are there: vegetative, asexual and the sexual means of reproduction. Let's see the heading first of all. What do you mean by vegetative? Sometimes we are feeling there is no difference right now in this under fungus. There is no difference between vegetative and asexual. If you go in a minute detail, right, then vegetative means the body of an organism is giving some part out and that is giving rise to new progeny. That is vegetative. In asexual means the parent body is forming some special structure which you call it as spores, but not by fusion of gamete, right, and not by meiosis. If it is happening by a fusion of some positive negative strains like gametes are there, they are going to fuse, then there is a meiosis in that, then spores are formed that are asexual spores. Getting that point? So there is a difference between vegetative, asexual and the sexual means of reproduction. Let's see under vegetative what comes. One is the fragmentation, where being it is filamentous one. In the living world chapter you will find this line that fungus even show this type of the reproduction, that is fragmentation, right? Even it can show the fusion of the cells, mostly in unicellular categories. Budding even, like in yeast, which is the most common example here, bud formation. You will find the bud chains over there. So this will come under the vegetative reproduction under the fungus. Now asexual one is what? It can be by conidia, sporangiospores, or even can by the zoospores. But here very important question lies. What is that? See the word very carefully. Zoo means which can move. Means they have locomotory structures over there like flagella. Means, or you can say flagella will be the locomotory organs. So can I say these spores will be motile? Right? Whereas these one will be immotile. Fine. First important question on this asexual means of reproduction. Fine. And next important on the same asexual type is what? When you will see these two. Now sporangiospore and zoospore. Its origin is endogenous. Endogenous means it is enclosed by sporangium. Like suppose if I am saying sporangiospore, it is enclosed inside a covering which is an extension from this sporangiophore. Sporangiophore, P-H-O-R-E. Phore means some filament, or you can say the hypha which is bearing that sporangium. That hypha you call it as sporangiophore. What do you call it as? Sporangiophore. Are you getting that? Now these sporangiospores are formed inside this structure which you call it as sporangia, or singular is sporangium. Are you getting that? So can you see it is enclosed inside some enclosed body which you call it as sporangium. Singular is sporangium. If plural is there, then sporangia. And the hypha which is bearing that hypha, that you call it as sporangiophore. And inside what is forming? Spores now. Which spores I am taking right now? Sporangiospores. Are you getting? Same thing happen with the zoospores. When it is forming, then they are having flagella on it. But it is enclosed inside the sporangium. Then you call it as zoosporangium. Clear? So sporangiospores and zoospores are endogenous in origin, whereas conidia, I am taking here now, suppose this is conidiophore. It is making conidia on its tip like this. Can you see here? It is outside this conidiophore. What is conidiophore now? Again, this is a hypha which is bearing the conidiospores, but not enclosed inside some enclosure, right? So they are outside. So can I say this conidia will be exogenous? It will be exogenous in its origin. Getting that? So please do remember these important points. I repeat, which one will be immotile? Conidia and sporangiospores. Which will be motile? Zoospore. That name will tell you. Zoo means moving. So rest two will be not moving, right? They don't have flagella for that. Now which one is endogenous in origin? Zoospore and sporangiospore. And which one will be exogenous in origin? Conidia will be there. Fine. Okay. Now coming to the sexual means of reproduction. It can be by oospores, ascospores, basidiospore. Our today's main discussion will be on the oospore formation, ascospore and basidiospore. And even there, the most important are ascospores and basidiospores to discuss. Reason why? From where it is forming, right? You will find the word basidium, basidiocarp. Likewise, you will find the word the SI and even the ascocarp. These words you will find. But what are these words? Right? You cannot able to figure out because NCERT book is not explaining with some diagrams that what is ascocarp, but what is basidiocarp, what is SI, what is basidium. To clarify that concept more, we have to discuss the ascospores and basidiospores in more detail, right? But you must remember oospore, ascospore, basidiospores comes under, or you can say these are the product for the sexual reproduction, right? So this is the layout of the means of reproduction. Now we are going to take the sexual means of reproduction in more detail. Let's see here what happens. Before getting into that, we must know about the classes. That how many classes are there in case of the kingdom fungi. Let's see the first one like Phycomycetes. It is there. No doubt here means of reproduction through sexual reproduction, right? There you will find the word oospores and even zygospores written in your NCERT book, right? But how it happens, we will discuss in the discussion. Right now, I am taking all the classes of the kingdom fungi, just overall view its name. So, classes next is the Ascomycetes, right? Where the sexual spores will be ascospores. And Basidiomycetes, obviously it will be the basidiospore. But very important question is that Deuteromycetes will not show the sexual reproduction. That's why you call it as imperfect fungi. Are you getting that? So this is imperfect. So definitely we are not going to take Deuteromycetes in our discussion, being it is not showing the sexual means of reproduction. Now let's take Basidiomycetes. No doubt it will show sexual reproduction, but problem here is that they are not forming sex organs, right? And one more important thing regarding that, they will not show a sexual reproduction. They will not show a sexual reproduction, right? Phycomycetes will show both asexual and sexual means of reproduction. Ascomycetes. Ascomycetes will show both sexual as well as asexual means of reproduction, right? Phycomycetes even will show the both, right? But do remember that Deuteromycetes will not show sexual reproduction, whereas Basidiomycetes will not show the asexual reproduction. Even no sex organ formation there, although it is reproducing by sexual means of reproduction. Then how it comes, that only we have to discuss through the detailed diagrams over there, right? Okay, and do remember, in all the things, what is common over there? That is conidia formation, right? It is very common in all, right? Except one or two exceptions. I am not listing that out. But conidia formation, vegetative reproduction is common here, right? So this is all about a outline regarding the classes. Now, why we classified them? On what grounds actually we have classified? Number one, based on the morphology of its mycelium. Now, what is this mycelium? When the hypha, which are filamentous thread-like structure I said you, which can be septate or aseptate, when they form network with each other or meshwork, that you call it as what thing? Mycelium. Like you are having a fabric, no? In that fabric, just take out the threads out, that you call it as hypha, or you can call it as hyphae, and complete fabric you call it as mycelium. In the same way here, in the case of fungus, you will find the complete network of the hyphae as mycelium, and each thread out of that you call it as hypha as a singular, right? And plural will be hyphae. Fine. So based on morphology of mycelium, second, even based on the mode of formation of the spores, right? Even it is based on the type of the fruiting body what they are forming. On that grounds, we are classifying them. Today's our topic is on reproduction of the fungus. So we will focus on its reproductive part. Now let's discuss about what is happening in its sexual means of reproduction. There the first step is happening as a plasmogamy. Second is the karyogamy. Third is the meiosis, right? Let's check out what is happening in all these steps one by one. In the first step, plasmogamy, as the word is indicating, that once the compatible type of the cells, or you can say reproductive structures, when they come close to each other, their cytoplasm or protoplasm is going to fuse, right? So that fusion of protoplasm of that compatible type, mating type of fungus, one will be positive mating, one will be negative, right? So just to say donor and the recipient, we call it as plus and minus. When that fuse, that you call it as plasmogamy. Gamy means fusion, right? Plasma means the things which are inside the cell except nucleus, that will not fuse, right? At the first step. At the first step, the nuclei of that two compatible structures will not fuse, only rest of the thing will fuse, right? So can I say the haploid cell or the structure and the haploid cell or the structure, they will fuse, that will in one cell to create a single cell which is having two nuclei inside that, right? Now in karyogamy, what happened? The word karyo means what thing? Nucleus. What N and N I have represented? Two nuclei inside one structure, one cell now. Otherwise, these are the two structures or two cells or two gametes which are there, and now they fused. Two nuclei are still there in one cell. This is the plasmogamy. When karyo means nucleus. Gamy means what thing? Fusion. When the fusion of these nuclei happen, then it will create a 2N structure. Can I say this will be the diploid structure now? Right? To get back this haploid structure again, which is a ploidy of the parental mating type, that will undergo the meiosis. You know that meiosis is a reductional process which will convert diploid again into haploid. Haploidy will come back in the form of spores. Now, where that spores will germinate, the new fungus will develop and we will get a new progeny of it, right? So, let's repeat what is the first step in the sexual means of reproduction? Plasmogamy. Second is karyogamy. Then third is the meiosis. Now, here do remember, there is a very interesting thing happens. Just now we discussed the various types of classes, Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes, right? Deuteromycetes is now out of our list. Reason we are discussing about the sexual means of reproduction, right? So, sexual reproduction is not shown by Deuteromycetes. If I discuss in case of Phycomycetes, what will happen? All these steps, one after the another, is happening immediately. Means there is no gap. Everything is happening immediately, right? But in case of Ascomycetes and in case of Basidiomycetes, what happens? Here, after the plasmogamy, the karyogamy is delayed. What is happening? After the plasmogamy, karyogamy is delayed. In between that, this plasmogamy and karyogamy, there is one more phase which you call it as dikaryon phase, in which this cell will have two nuclei, which is haploid and haploid. Di means two, karyon means nuclear. Two nuclei will be there. Nuclei is a plural of nucleus, right? So two nuclei are there. They are still not fusing. That stage you call it as dikaryotic phase. That phase you call it as dikaryotic phase. So in which case the dikaryon phase is there in case of Ascomycetes and Basidiomycetes. Which phase is there? Dikaryon phase, or you call it as dikaryotic phase, right? So in which case, I repeat, Ascomycetes and Basidiomycetes. If they give you in few of the fungus, then Phycomycetes. If they are saying most of the fungus, then do remember we are talking about Asco and Basidio, because two classes included there. If they are not mentioning Asco and Basidio, maybe they are giving you in the question the most of the fungus is showing which stages. Then your answer should be plasmogamy, dikaryotic phase or dikaryon phase. After that, no doubt karyogamy will happen, but it will be delayed. It is not immediately happen after the plasmogamy. After that will be meiosis. This is all happening for Ascomycetes and the Basidiomycetes. I hope so. This is fine to everyone. Now, if I take what are the stages of sexual reproduction in Phycomycetes, then your answer will be plasmogamy, karyogamy, meiosis. Immediately, one after the another. But there is no dikaryotic phase, right? So, this is about Phycomycetes. Now, I am taking next about the Ascomycetes. What is happening there? Now, your main confusion, I hope so, is going to be solved here when I will discuss with you what is SI, what is Ascocar, and what are ascospores, right? What are the differences among that, right? Let's check it out. This is a sexual reproduction in case of Ascomycetes. And don't go after full diagram at a whole. What I am pointing, just focus on that thing. I hope so it will be clear, right? Now, if I am taking this discussion in detail here, you will find in the hyphae. I repeat, this is the hyphae of that, right? Which hyphae it is? That is a primary hypha. Keep in your mind, this is which hypha? Primary hypha, where you are having their own individual nuclei, right? Now, they are having sex organs, which is clearly mentioned here. When you will check here, can you see they have shown you antheridium and ascogonium categories? Antheridium is the male structure. Ascogonium is a female structure, right? One thing must be clear, which is male and female. Antheridium is the male sex organ right now. Ascogonium is the female one, right? And now what happened? These two hyphae are the original hyphae, or I must say first stage of the vegetative fungus, which start undergoing the sexual reproduction, having the female and male sex organs during their sexual reproduction. Actually, they will propagate more easily in warm and humid places, which is even one of the characteristic feature of the fungus. They need warm and humid places. When it will be coming, then it will become more easy for the fungus to show all these processes. So, let's repeat. This is their primary mycelium, which is having mating plus and mating minus. Plus is going to receive, minus is going to give that. Now, see here, this will be the plus, this will be the minus. Blue one is the minus and red one will be plus. So, plus is taking from the minus. I repeat this point again. Minus will give that, plus will receive. As per here, minus is the blue and red one is the plus. So, minus is giving to the plus. Are you getting that? So, where the thing is added, that is a plus mating type, which is giving, that is a minus mating type, being it is giving to the positive strain. Just to simplify, they are not giving exactly the word male and female bodies. They are simply indicating by plus and minus sign. That's it. You need not to worry who is taking, who is giving that. It is not your job. But keep in your mind, the ascogonium is a female which will receive the thing from the antheridium, which is a male. Now what will happen? See next. There is a connection which is going to build between antheridium and ascogonium. Now due to that, all the nuclei from here will enter into ascogonium. Now each cell of that, where this is receiving the extra nucleus, that instead of one is having two. Let me show you in the previous slide what I have discussed, that one cell haploid, second cell haploid will fuse to give one cell. I am talking about one cell. They have shown many cells in the diagram. So come out with the result. Can I say here at the last, we are getting some structures which is having two nuclei here. Can I call it as dikaryon stage? Are you getting that? What do you call it as a dikaryon stage, having the two nuclei in the one cell. Fine. Now what will happen? This dikaryon stage will be on some mycelium, right? This will be on some mycelium. What is that mycelium? Which is the primary mycelium of that. What is primary mycelium? This one, right? This is the primary mycelium which has given out this sex organs, and that sex organ make single nucleated structure as a dinucleated structure. Just that keep in your mind. Fine. It is a dikaryon stage. Now what will happen? This cell will undergo mitosis, right? Can I say every cell is now having two nuclei? They will keep on dividing mitotically and give you this cup-shaped structure, which you call it as a ascocarp. What do you call it as? Ascocarp. Carp means fruit. It is like a fruiting body for the fungus. What do you take it as? Ascocarp. It can be cup-shaped, it can be globo-shaped, it can be cup and saucer shape, or correct word I must say, it is like a, you can say like a flask shape as well. Cup shape, flask shape, cup and saucer shape. These are the three main shapes of ascocarp. What is ascocarp? Fruiting body of ascomycetes. How it have been created? From a cell which is carrying two nuclei, it repeatedly undergoing mitosis. So every cell is having two nuclei. So can I say it fruiting body? Every cell is having two nuclei. Keep that in your mind, right? Now what will happen? Suppose they are taking only one cell out of it in the ascocarp at its tip. One cell which is having two nuclei. Can you see two nuclei they have shown here? Every cell is now having two nuclei. Whatever you will take it out from that. Now what will happen? One of the cell, here the fusion of nuclei happen. That fusion of nuclei happens over there only in one of the structures, but at the usually periphery, not in all, right? At the periphery, when the fusion of this nuclei happen, can I say it will give rise to diploid structure? And this stage you call it as what thing? Karyogamy. It is going to be delayed. Why it is delayed? Because here this stage is extra in ascomycetes, which is forming the fruiting body. Right? In fruiting body, every cell will carry the two nuclei. And can I say this is a fruiting body which is on this primary mycelium. Primary mycelium will degenerate. Now in nature, what is left? A fruiting body. Right? And in fruiting body, especially at periphery, if we will take, what will happen? Here the fusion of nuclei will happen and give rise to this diploid nucleus. And where this fusion happen, that cell you call it as ascus, or plural you call it as SI. What do you call it as in plural? SI. Singular, what do you call it as? Ascus. Right? Have you understood the difference between ascocarp and ascus? Ascocarp is a fruiting body which is secondary mycelium. Every cell is having two nuclei. It is start forming on primary mycelium, parental mycelium. But parental mycelium will degenerate slowly. You will get in nature only this fruiting body. And after that, SI is present over there. Where karyogamy happen, you will get diploid thing. That diploid will undergo meiosis and forming. Obviously, you know that one nucleus will give rise to four nuclei after the meiosis. Are you getting that? So, can I say the correct thing? Each nucleus obviously is enclosed inside the cell. Here that cell, you call it as ascospores. Meiosis 1 will give 2 cells which are haploid. It further undergo meiosis 2 which will give rise to 4 cells at last. That 4 cells you call it as ascospores. Now can you see here 4 cells have been created? That cells you call it as ascospores. All the material will be clustered and will form the new cells within this ascus, right? Ascospores have been formed. As it is formed by meiosis, so its ploidy will be haploid, right? Now even you can call it as meiospores, being it is formed by the meiosis. Fine. Now what is happening? Special thing here, ascospores, it will undergo again mitosis. I said you, it is formed by what thing? It is formed by meiosis. It is haploid. How many form? 4 form. But after mitosis, each will become 2. Can I say total ascospores at last will be forming 8. How many ascospores will form at the last? 8. Actually form how many in the beginning? 4. Now each one undergo mitosis. How many we will get? 8. And can you see it is still enclosed inside this structure? That's why it is endogenous. The very important word here is the endogenous. Are you getting that? So can you see it is enclosed inside the body here? And that's why I am writing opposite to that. Now it is very important to write here. They are endogenous in origin. Fine. Once this stage have been reached, the fruiting body get mature, it will burst. And all the spores are going to release. Again giving rise to this mating type, right? Don't go after that positive, negative much. Just remember in your mind, in ascomycetes, they are forming sex organs, right? And again, the cycle will start like this. This is happening in case of ascomycetes. There is a plasmogamy. This step is what thing? Plasmogamy. Are you getting that? And here what have been formed? Fruiting body, dikaryotic phase. Can I say karyogamy have been delayed? Here they are going to fuse haploid nuclei at this point. And this step you call it as what? Karyogamy. Right? Due to the dikaryotic phase, karyogamy has been delayed. And after that, what happened? Meiosis happened. Right? So this is about the ascomycetes. Now let's see what is happening in basidiomycetes. Number 1, this is the primary mycelium. Like in earlier, the parental mycelium, I said you, primary mycelium, which every cell is having single nucleus. Like earlier even, this is in ascomycetes, every cell is having single nucleus. Are you getting that? Here even, single cell is having single nucleus. But there is no formation of sex organs. Just now I discussed with you. Right? So, that type of fusion you call it as somatic fusion. Right? Or you can say vegetative cell are going to fuse here. Means their hyphae cell will be fused of the compatible hyphae of the mycelium. So once they are fusing, suppose I am taking this one is getting fused here. Right? Rest of all are going to be much of the unfused type, but few are going to be fused. I am taking one only here, right? I am not taking rest of that. I am taking one. So, one of that is going to be fused. It can be more than one can fuse as well. But right now, as per this diagram, you have to consider only one have been fused like this. Now, this one will undergo mitosis and give rise to this extension, the new hypha. Can you see in this new hypha, 2 nuclei will be there? Are you getting that? So here, once the fusion happen in somatic cell, you will find the dikaryon stage. And that dikaryon stage keep on multiplying by mitosis, right? So this dikaryon stage will keep on multiplying by which process? Mitosis. And at last, you will get this type of the fruiting body. Now here, fruiting body, you call it as basidiocarp, which is a dikaryotic phase. Can you see the mushrooms? What you see in the nature? It is the basidiocarp, fruiting body of basidiomycetes. At the below surface, you will find gills. which is having foldings on it, like this. Foldings are there. Can I say each folding is having 2 nuclei? Because this is a feature of this full structure, right? This even you call it as secondary mycelium, right? Now every cell is having the 2 nuclei. Now what will happen over there? These every basidium, every part, every such type of bulging part, you call them as what thing? You call it as a basidium. Singular as basidium, plural as basidia. Such type many will be there. This is equivalent to SI in ascomycetes, right? And basidiocarp is equivalent to ascocarp there, right? So now here in basidium, what will happen, or basidia, there will be the fusion of the nuclei are going to be there. When nuclei are going to fuse, that process you call it as karyogamy. Can I say we will get diploid cells now here? That diploid cell will undergo meiosis. And here one basidium is forming how many haploid nuclei? Or the cells inside that 4, which further will make the spores with a proper structure over there. So means at last we are getting on one basidium. How many spores we are getting over there? 4 spores, like here. You will find here, like 4 spores are there. It will not undergo mitosis to form 8 now. And can you see it is not enclosed inside the covering? It is outside. That's why the basidiospores are exogenous in origin. Whereas in the ascomycetes, it will be endogenous. And per ascus, it was 8. But here it is exogenous in origin, and in number it will be 4. Getting that? Now last, let's see the difference. Ascomycetes, basidiomycetes. Here there is a formation of sex organs. But here there is no formation of sex organs. Right? And here you will get cup shape or cup and saucer shape, globo shape or flower shape type of ascocarp. Here you will get like a mushroom umbrella shape type of the basidiocarp, which is a fruiting body. Here you will find at the last ascocarp is endogenous. Here basidiocarp is going to be exogenous. This is exogenous and this is going to be endogenous. Here ascocarp endogenous, I must say ascospores, sorry. Ascospores will be endogenous. Here basidiospores will be exogenous. Fine. And at last, how many you are getting in number? 8. Here you will get 4. Fine. So this is very important differences that you should know. And here you are having ascus and here you are having the basidia as a plural, basidium as a singular. Fine. So these are the means of the sexual reproduction in the fungus. Right? The main logic, you should know what is basidium, what is SI and what is the ascospore, basidiospores, that you should know, right? And what is a parental type of mycelium, what is secondary mycelium. The carps or fruiting body are secondary mycelium, which is having 2 nuclei, whereas parental one is uninucleated. Each cell is having 1 nucleus. It is a start of the journey of sexual reproduction in case of the fungus, like in case of ascomycetes and basidiomycetes, just now what we have completed, right? So this is all about today's discussion. I hope so. Some clarity will happen in this topic. Just go through once, you will get more about it with the examples. Thank you.